Hi, Dr. Brian Kaufman here at ASH 2023 as the Chief Medical Officer, Executive Vice President of the CLL Society.
And I'm Alexey Danilov, Professor in the Department of Hematology and leader of the Lymphoma Center at City of Hope in Los Angeles, California. Pleasure to be here.
Dr. Danilov, we've talked a little bit about a new way to block BTK with degraders. There's a new degrader trial that you're starting at City of Hope that you're involved with, a different molecule. Can you talk a little bit about that and why you're excited about this trial, and if there's any early data? I know it's very, very early, but if there's any early data, tell us a little bit about that molecule, why you think it might be helpful to a CLL patient.
Yeah, this molecule is something called a PROTAC, and that's an abbreviation that I believe we'll hear quite a lot in the next few years. PROTAC stands for proteolysis targeting chimeras. A simpler name for that molecule is a BTK degrader. And as you know, Bruton tyrosine kinase, or BTK, is very important in pathogenesis of chronic lymphocytic leukemia. And as we've learned in the past 15 years, CLL cells are so dependent on BTK, which links them to the supportive environment in the lymph node, that we can disrupt it with some of the small molecules, which we all know about now, which we have used consistently in our practice, such as ibrutinib, acalabrutinib, and zanubrutinib. The trouble is that eventually the majority of patients, even though they respond for a very long time and have minimal toxicities, develop resistance to these BTK inhibitors. And the resistance is typically driven by mutations or alterations in the genetic sequence of this particular protein kinase, Bruton tyrosine kinase, BTK. So this is becoming an unmet medical need. And as you know, pirtobrutinib, a non-covalent BTK inhibitor, has been recently approved in therapy of CLL for patients who have been exposed to BTK inhibitor or BCL2 inhibitor. However, resistance mechanisms to that drug are similar, where mutations still occur and CLL becomes refractory to all BTK inhibitors, essentially. So BTK degraders use a completely novel approach. Instead of binding to the BTK pocket, ATP pocket, and inhibiting its function, they actually grab it and shuttle it to the proteasome. And proteasome is like a little garbage truck which is responsible for destroying anything that cell doesn't want anymore. So as a result, BTK gets processed down the chute and gets expelled from the cell as waste. And it's no longer there, and therefore its function is, by definition, is inhibited. But because these protein degraders or BTK degraders don't care so much whether BTK is mutated or not, they don't care what kind of a flower is drawn on the BTK or whether there is a little decoration or not, or something sticking out, which the mutation typically would change the conformation, how the kinase looks. They are effective they're effective against CLL cases who became resistant to all BTK inhibitors.
So there's a new molecule that you're looking at that does this degrading. Do you have, tell us a little bit about it specifically, and do you have that, I think it's 5948, did I get that right?
That's right, yeah.
NX5948. So you have a trial open now at City of Hope with this. Tell us a little bit about the trial and if there's any data there in terms of how it's working.
Yeah, so it is NX5948. It's a BTK degrader which is made by Nurix, a biotechnology company which specializes in this technology. And the trial is still very early. It's a phase 1 trial, which is currently in so-called dose escalation phase, meaning we slowly escalate how many milligrams a patient gets of this drug every day to find out what the toxicities are and what the efficacy is. And of course, the primary thing we care for is toxicity initially, to make sure that there are no big issues associated with the drug. So currently, there are about 13 patients on study. Roughly half of them have CLL, and we progressed through 3 dose levels. And so far, it is very impressive that there have been no major toxicities. So we progressed through these dose levels very quickly. And it's an oral agent. It's a pill which patients take once per day, very similar to ibrutinib. And we progressed through 50, 100, and 200 milligrams per day without any significant toxicities, which we call dose-limiting toxicities. At the same time, we do see degradation of BTK when we draw the cells from patients' blood and analyze BTK. We do see its disappearance once patients start on the drug. So the mechanism is proven, yeah? So we have proof that this drug actually works mechanistically. Not only that, we do see that the drug works clinically. We already see partial responses with this particular agent. As you know, with any drugs which target BTK, it takes a while to get the so-called complete response, which is full disappearance of all lymph nodes and full recovery of all blood counts. That can take years with ibrutinib or similar drugs. So even the fact that we see responses, even if partial, so early in the trial are very encouraging when we're just using such small baby doses of this drug.
This sounds very encouraging. So tell us, if patients are interested in this trial, who would be a good patient for this, and how can they find out more about the trial?
You know, the majority of patients who we enrolled so far have received, have been treated with a BTK inhibitor such as ibrutinib, zanubrutinib, or acalabrutinib, and venetoclax. But even patients who just had received only BTK inhibitor, they may or may not have received pirtobrutinib. As long as they received 2 lines of therapy of some sort, they would be eligible for the trial. You know, there are some standard eligibility criteria, some drug interaction, some potential drug interactions that we have to pay attention to depending on patients' other medications. But the trial is open and enrolling, and the efficacy is really good. And the other thing I want to mention, responses are very quick. Within a week or 2, we already see improvement in lymphadenopathy, and even improvement in large lymph nodes, and even improvement in low red cell counts in some patients who come with this problem to us. So the clinical benefit is very early.
This is at City of Hope. Is it elsewhere, or are you the only site at this point?
This is at City of Hope. There are other sites as well. So on clinicaltrial.gov, there would be a full list of sites. So you can search it by term NX-5948 in the other category. There is a small search engine, so you would be able to see the sites. There are a lot of sites in the United Kingdom. That's where the study was initially open, and now it has been expanded to the United States.
Any final thoughts on the degraders or NX, the new NX-5948 and how that works?
Yeah, so I think it's a very exciting field of new molecules. I think it's becoming a class of agents which we will hear more and more about. I'll just make a comment that if you think of this mechanism, you can also target other kinases or proteins, not just BTK, but some undruggable proteins that we have been working on for a very long time. For example, MYC, which can be relevant to CLL or Richter's transformation, and other proteins, even BCL2, other BCL2 family members. And there is a lot of chemistry research going on now, how we can make these new molecules. But for now, to get back down to earth, BTK degraders is a very promising new class of medications, which I believe will reshape how we treat CLL, which progresses on BTK inhibitors.
Well, I'm very excited about this. I'm very grateful for the research you and your colleagues at City of Hope are doing. We'll put up a link to this trial so patients can find out more about it. Thank you so much, Dr. Danilov.
Thank you.
Thanks.
Thank you, Dr. Kaufman.